Okay. Welcome to the CVRTI Seminar Series. Just as a reminder, we've got several external speakers next month. Emails will be going out about that, and invitations for meeting both with trainees and faculty with our external speakers. So look for those coming up. Today we're delighted to have Lisa Lesniewski with us today. She, by way of introduction, did her bachelor's at Ohio State University in exercise physiology, and she did a master's at Kent State after that, before going to Texas A&M to do a PhD in exercise physiology. And then she did postdoctoral training at UC San Diego and University of Colorado Boulder. After her PhD or postdoctoral training, she came to the University of Utah and has been here since 2010. Has a lab at the VA and also an appointment here in internal medicine and geriatrics, and was an assistant professor and then associate professor. Her research is funded by a number of different agencies, NIH. She has a VA merit grant. It's a merit, right? As well, yeah, merit grant, and had funding from other institutes, other groups like AHA and Kidney Foundation, I believe. At some point, no, not kidney, not kidney. Okay, got it wrong. Anyway, she co-directs the Vascular Translational Physiology Lab at the VA, and really brings an important factor here to our discussion at CVRTI with the vascular research. And delighted to have her here to speak with us today. So she'll give her talk, and then we'll have some time at the end for questions from the audience. Lisa, thank you.
Thanks. Okay, well, thank you for the invitation to come and present today. What I want to talk about is some work that we've done over the past few years that really started with examining the impact of aging on vascular dysfunction and the mechanisms that underlie that. And then some findings that came out of those studies that really implicated changes in endothelial function and the maintenance of metabolic homeostasis. So first, we know that cardiovascular disease remains among the leading causes of death. And this is true for both men and women. And you can also see that diabetes, we can also see that diabetes is among the leading causes of death as well. And we know that aging is a primary risk factor for both cardiovascular and metabolic diseases. And you can see here in this graph that there's an almost linear increase in the prevalence of both cardiovascular disease and diabetes with aging. And it's been appreciated that dysfunction in the vascular endothelium is an important mediator of this increase in cardiovascular disease risk, and is also a common comorbidity of metabolic disease. So first, I just want to give a brief overview of what the endothelium is. So the vascular endothelium is just a single layer of cells that lines the inside of our blood vessels. And when it was first described, it was thought to be an inert barrier that just separated the circulation from the organs. And it's now known to be a dynamic organ that not only controls the barrier through changes in permeability, but also plays a role in the maintenance of blood in a fluid state through control of thrombosis. It participates in the formation of new blood vessels through the process of angiogenesis. And it controls vascular tone through changes in vessel diameter through vasodilation and vasoconstriction. And this is important because it will then determine tissue blood flow as well as participate in the regulation of blood pressure. So the endothelium acts as both a sensor and effector. It can sense hemodynamic and mechanical forces such as shear and stretch. It also is the site of interaction with circulating factors such as cytokines, immune cells, or other drugs that are in the circulation. The endothelium itself also secretes vasoactive and metabolically active substances, as well as cytokines and reactive oxygen species. And these things can act back on vessel function, but can also act on the surrounding tissues to alter their function as well. One of the primary factors that are secreted by the endothelium is nitric oxide. Nitric oxide is produced by the endothelium in response to shear stress or vasodilatory agonists. And in addition to being a potent vasodilator, it also acts in an antithrombotic, anti-proliferative, and anti-inflammatory manner. And we know that with aging, there's a reduction in nitric oxide bioavailability that will lead to impairments in endothelial function in general, and that this contributes to the increase in cardiovascular disease risk that we see with aging. Underlying these changes with aging, a number of processes become dysregulated. So among these are changes in growth factor signaling, nutrient sensing pathways, and genomic instability. When these things become dysregulated, we'll see changes in cellular function that increase cellular senescence, as well as dysregulate autophagy. This can then contribute to increases in inflammation and oxidative stress that then lead directly to impairments in endothelial dysfunction, as well as increases in our arterial stiffness that then contribute to cardiovascular disease. So among these things that become dysregulated with aging, we're interested in the mTOR signaling pathway. So what is mTOR? This is a ubiquitously expressed serine threonine kinase that exists in two complexes. The most highly studied of which is the mTORC1 complex. This is also the one that is most sensitive to inhibition by rapamycin. mTORC1 is sensitive to changes in growth factors and amino acids, as well as oxygen and energy status. And this will lead to increases in the synthesis of proteins, lipids, and nucleotides, as well as the inhibition of autophagy. And we became interested in mTOR after seminal work that was performed by Richard Miller's group, in which treatment of old animals, or actually middle-aged animals, with dietary rapamycin could lead to an increase in lifespan in both male and female mice. So from this, we were interested in seeing what happens with mTOR activation in the arteries with advancing age. So here we're looking at young and old mice, so 6-month and 24-month-old mice. And we wanted to examine endothelial activation of mTOR with aging. And so here you see end-phase staining of the aortas of young and old mice. And there is a significant increase in phospho S6K, which is a downstream target of mTOR activation. And we see this both in the vascular smooth muscle and the endothelial cell layer, but it's a much greater increase at the endothelium. So we published back in 2017 some work examining the effect of dietary rapamycin on endothelial function. And I just want to show you a little bit of that. But first, I'm going to explain how we measure endothelial function in our mice. So the example here is a mesenteric artery that's been excised and cannulated onto 2 glass micropipettes that are in the bath of this pressure myograph. We can apply drugs or shear stress through the lumen of the vessel, and then examine changes in lumen diameter and generate a dose response curve that would look something like this. If we want to examine the impact or the role of nitric oxide in this vasodilation, we can apply a inhibitor of nitric oxide synthase called LNAME to the bath, and then repeat this dose response. And when we do so, the difference in dilation in the absence or presence of LNAME gives us our measure of nitric oxide bioavailability. So this is what we did in mice treated with rapamycin in the diet. And we used the same diet that was used in the Richard Miller study that was shown to increase lifespan in mice. And so you can see in the closed circles here, the response to acetylcholine for dilation from a young normal child-fed animal. And when we apply LNAME to the bath, we see that about 50% of the dilation is due to nitric oxide. With aging, we see a significant reduction in dilation in the carotid arteries of mice. And this results from a reduction in nitric oxide bioavailability. When we fed the animals a rapamycin supplemented diet, we saw no effect on dilation in the young animals. However, Well, which way do I go? Shoot, sorry. We saw a significant increase in dilation in the old animals when fed the rapamycin supplemented diet, and this resulted from an increase in nitric oxide bioavailability. Interestingly, we also performed glucose tolerance tests in these mice. And to do this, what you do is fast mice in the morning, you take their blood glucose under the fasted state, you inject them with glucose intraperitoneally, and then monitor their glucose over the course of 2 hours. In this case, I'm showing the data for the area under the curve. If there's a greater area under the curve during the glucose tolerance test, that would indicate glucose intolerance or metabolic dysfunction. So what we saw when we examined glucose tolerance in the young and old animals treated with rapamycin was an improvement in glucose tolerance in the aged animals, but a deleterious effect in the young with a very large increase in the area under the curve. So an impairment in glucose tolerance. So from this, we became interested in determining if we could knock out endothelial mTOR and derive the beneficial effects on the vasculature without getting the deleterious effects on metabolism. So to do this, we generated an tamoxifen-inducible endothelial-specific mTOR knockout animal. We did this using the CDH5 or VE cadherin Cree animal that we obtained from Rolf Adams, and then the mTOR flox animal that we got from Matt Rondina. And you can see here, we again did the end phase staining of the endothelium to determine the impact on mTOR expression after tamoxifen treatment in these animals. And we could see about a 75% reduction in mTOR expression in the endothelial cells of these animals. So we first wanted to examine the effect of mTOR deletion on endothelium-dependent dilation. So you see here in the red circles, the response in the wild type animals to acetylcholine, and in the blue squares, the response of the endothelial mTOR knockout animals to acetylcholine. In our young animals, we saw no effect of mTOR deletion on vasodilation or on nitric oxide bioavailability. However, when we examined dilation in old arteries, we saw a significant decline in dilation in the mesenteric arteries of the aged animals for wild type mice, and a small but significant increase in dilation after mTOR was deleted in the endothelium. And this resulted from an increase in nitric oxide bioavailability after the loss of endothelial mTOR in the aged mice. And we wanted to make sure that these changes were due to changes in the endothelium and not the vascular smooth muscle. So we performed endothelium independent dilation in response to sodium nitroprusside, which is a nitric oxide donor. And we could see no differences in dilation to this NO donor between young and old animals or wild type or endothelial mTOR knockout. So next we wanted to see what might be mediating the increase in dilation in the old animals after mTOR is deleted in the endothelium. So first we assessed superoxide, and this is in an old carotid artery via electron paramagnetic resonance. And we could see a significant reduction in superoxide content in the carotid arteries of the mTOR knockout animals. And to make sure that this is actually mediating the changes in dilation that we observed, we can perform the dose responses to acetylcholine in the presence of a superoxide dismutase mimetic, in this case Tempol. When we did this in the young or in the old wild type animals, again in those red circles, we can see a restoration of dilation in the presence of Tempol, indicating a superoxide mediated suppression of dilation with aging. And again, when we looked at the mTOR deleted animals, we already saw an improvement in dilation in those blue squares. And with Tempol, we saw a further improvement in dilation. So these results suggest that the superoxide reduction after mTOR deletion is what is mediating that increase in dilation. And next, we were interested in exploring if this also was related to changes in senescence and inflammation, as we know that inflammation and oxidative stress act in sort of a feed-forward circle to induce endothelial dysfunction in aged animals. So here we took the thoracic aorta of old animals of both wild type and knockouts, and we used markers of, I don't know if I can point to anything, but markers of immune cells as well as inflammatory markers, and P16 and P21 are markers of senescence. And we could see across the board that deletion of mTOR from the endothelium was able to reduce markers of both senescence and inflammation. And so this suggests to us that the endothelium may be having a less inflammatory, less secretory phenotype after mTOR is deleted, and that this could act not only to improve the function of the vessels themselves, but also to act to improve function of the surrounding tissues. And that might include metabolically active tissues such as the skeletal muscle, adipose, and liver. So we next wanted to ask the question, what the impact of endothelial mTOR deletion was on metabolic function in these animals. So to give, oh, there's a weird typo in there. So to give a very quick and overly simplistic view of glucose and insulin metabolism, just so we're all on the same page. When glucose is in the circulation, it is sensed by the pancreas that then releases insulin from the beta cells. That insulin then travels back through the bloodstream to the adipose and the skeletal muscle where it induces glucose uptake. At the adipose, it also inhibits lipolysis, and at the liver, it inhibits gluconeogenesis. The actions of all of these things lead to reductions in glucose and free fatty acids in the circulation. In states of metabolic dysfunction, such as occurs with aging, you see an impairment in these insulin actions. So you get an increase in lipolysis and a decrease in glucose uptake.
The doctor will tell you when.
That adipose tissue. You also get a decrease in glucose uptake in the muscle and an increase in gluconeogenesis. All of these will lead to increases in free fatty acids and increases in glucose. And at the level of the pancreas, you will get a compensatory increase in insulin secretion in order to try to bring blood glucose back down to homeostasis. So when we examine metabolic function in our mTOR deleted animals, we saw the same improvement in glucose tolerance in the aged animals that we saw after rapamycin inhibition. However, in the young animals where we saw a large deleterious impact of rapamycin on metabolic function, there was no effect in glucose tolerance in the young animals. So from this, we can see that we were in fact able to influence metabolic function by influencing the endothelium specifically. And next, we wanted to determine which tissues might be involved in this beneficial metabolic effect that we saw in the aged animals. So to do this, we performed a battery of metabolic tests. First, we assessed glucose-stimulated insulin secretion. So here on that lower panel, you can see in the basal state, we measured blood glucose after fasting, and then mice were given glucose intraperitoneally, and we examined insulin in the plasma after 15 minutes. And you can see in the red bars, the characteristic increase in insulin in the plasma after this glucose challenge. And this was unaffected by mTOR deletion in the endothelium, shown in the blue bars. So next, we wanted to see if changes in insulin tolerance or insulin sensitivity could explain the differences in glucose tolerance. And so to do that, we performed an insulin tolerance test. And here we have fasted animals again that are now given an injection of insulin, and then blood glucose is again assessed over 2 hours. If you have a more insulin sensitive animal, you will see a larger decrease in blood glucose during the ITT. However, when we performed this in our wild type and endothelial mTOR knockout animals, we saw no effect on insulin tolerance, which suggests that there was not an impact on skeletal muscle insulin sensitivity, as that primarily drives the glucose uptake during this test. We next wanted to assess the effect of mTOR deletion on lipid metabolism. And to do that, we performed a lipid tolerance test. In this case, we measure fasted plasma triglycerides. Animals are given a bolus of lipid via oral gavage, and then we monitor plasma triglycerides over 3 hours. The lower the plasma triglycerides during the test, the better the lipid metabolism. And we could see here a significant effect of endothelial mTOR deletion on lipid metabolism. And this may result from changes in lipid uptake, lipogenesis, or beta oxidation. And that's something that we still need to work on elucidating the mechanism. And last, we performed a pyruvate tolerance test to assess gluconeogenesis in the liver. So this is done similarly. We're measuring blood glucose after an injection of pyruvate, which is a substrate for gluconeogenesis. And what we could see is that the endothelial mTOR knockout in the old animals resulted in a reduction in gluconeogenesis. So it appears that the improvements in glucose tolerance that we're seeing are largely driven by changes in metabolism within the adipose and the liver. And so we wanted to see if the changes we saw in senescence and inflammation in the endothelial cell were also found in these two tissues where we saw changes in function. And indeed, we saw almost across the board a similar reduction in our inflammatory and senescence markers in both the liver and the adipose tissue. And so to tie this all together, what we see is that if we delete mTOR in the endothelium of aged mice, we can improve endothelium-dependent dilation and nitric oxide bioavailability through reductions in reactive oxygen species that's also associated with reduced senescence and inflammation. And this then results in improvements in glucose tolerance that are associated with improved metabolism within the adipose and the liver. And this is also associated with reductions in senescence and inflammation. And so it appears that we really can influence metabolism by improving endothelial function in aged animals. But we were interested in seeing if the opposite was also true. Can we induce endothelial dysfunction and then drive impairments in systemic metabolism? To explore this, we assessed the impact of ARF6 on endothelial and metabolic function. So why did we pick ARF6? This is ADP ribosylation factor 6. It's a ubiquitously expressed small GTPase involved in a variety of cell functions. Its activity is regulated by GTP binding under the actions of guanine exchange factors and GTPase activating proteins. And it's been shown to have effects in both metabolically active tissues as well as the endothelium. So within the metabolically active tissues, we know that ARF6 participates in glucose-stimulated insulin secretion from the beta cell. It also participates in GLUT4-mediated glucose transport within adipocytes and is a modulator of hepatic insulin sensitivity. Whereas within the endothelium, ARF6 is known to participate in migration, angiogenesis, and permeability downstream of growth factor signaling. It also participates in permeability downstream of inflammatory cytokines, as well as in the co-receptor binding and internalization of growth factors such as VEGF. What's not known is if ARF6 actually plays a role in endothelium-dependent dilation. So that's the first question we wanted to ask. To do this, we again used the CDH5 or VE cadherin Cree animal and crossed that with a floxed ARF6 animal that we obtained from Wendy's Zoo. And after induction of by tamoxifen of this gene, we could see a significant reduction in gene expression for ARF6 within primary lung endothelial cells that were isolated from the animal. So that's shown on the far end over there. We also saw a smaller but significant reduction in ARF6 expression within the lung, but this is a highly vascularized organ, so it's not that unexpected. However, in the other tissues that we examined, such as the liver, skeletal muscle, adipose tissue, and kidney, we saw no effect of tamoxifen treatment on ARF6 expression. When we next wanted to examine protein expression, we used those isolated primary lung endothelial cells, and performed a Western blot and could see again about a 75 to 80% reduction in R6 expression within the endothelium from this model. So first, we wanted to examine dilation to see if R6 was able to modulate dilation in these arteries. And so because we're interested in the downstream effects on metabolism, we chose to perform dilation in response to insulin, as well as from arteries from a metabolically active circulation. So in this case, we're showing the gastroc feed arteries and increasing doses of insulin on the x-axis. You can see in the wild type animals shown here in the black circles, there is about a 30% dilation in response to insulin in these arteries. And after endothelial R6 deletion, this is significantly reduced. And this reduction is due to a loss of nitric oxide bioavailability. So those differences in the arrows are shown in the bar graph next to the dose response. Now, interestingly, this was specific to insulin and not a general defect in endothelial function, because when we performed dilation in response to acetylcholine, we saw normal function between the wild type and the knockout animals, as well as normal nitric oxide bioavailability. Likewise, when we performed the endothelial independent dilation in these arteries, we saw no defect. So this appears to be a very specific decrease in dilation in response to insulin. So next, we wanted to see if that is sufficient to impair metabolic function in these animals. So first, we performed a glucose tolerance test. And initially, we're like, oh, I guess that didn't work, because we saw absolutely no difference between the wild type and the endothelial R6 knockout animals for glucose tolerance. However, when we looked at the glucose-stimulated insulin secretion, we first saw that the R6 animals had an initial increase at the baseline in insulin in the plasma, and that upon glucose stimulation, this increase was even greater. And so this would suggest that there is a change in peripheral insulin sensitivity, and the increase in plasma insulin is compensatory to this. So to examine that, we performed the insulin tolerance test again. And here we saw a very large decrease in insulin sensitivity in the skeletal muscle of the endothelial R6 knockout animals. And like I said, this is largely driven by skeletal muscle, but you don't necessarily know that for sure. So we'll perform further testing to sort of elucidate that more fully. We next wanted to make sure that the changes we were seeing in metabolism weren't just due to changes in adiposity. And indeed, we didn't see any change in body mass or tissue mass for the skeletal muscle or the adipose tissue in our knockout animals. So to further explore the mechanism underlying the insulin intolerance that we saw, we performed euglycemic hyperinsulinemic clamps. So these are the gold standard for assessing glucose and insulin metabolism. They allow for an assessment of both peripheral insulin sensitivity, as well as for an assessment of the ability of insulin to suppress gluconeogenesis. To do this, you cannulate mice in the jugular vein to allow for the infusion of both insulin and glucose. Before you begin the clamp, you infuse tritiated glucose that will equilibrate in the body, and that will allow you to make this measure of hepatic glucose production, which is your measure of gluconeogenesis. At the start of the clamp, you begin an insulin infusion that stays at a constant rate throughout, and then you begin a glucose infusion at a rate that you modify in order to achieve a steady state glucose of about 120 milligrams per deciliter. The rate of glucose infusion at that steady state at the end of the clamp is the measure of peripheral insulin sensitivity. And then using the blood at the end of the clamp, you can also calculate this gluconeogenesis from hepatic glucose production. So when we performed this test, we were able to see a significant decrease in the glucose infusion rate, indicating peripheral insensitivity or peripheral insulin resistance in these animals. To see if this was related to changes in hepatic function, we calculated the glucose production and saw no significant effect on glucose production. So this would suggest that the changes in insulin sensitivity are largely being driven by the skeletal muscle. And so to summarize that, what we see is endothelial R6 deletion leads to reductions in insulin-stimulated dilation in the skeletal muscle arteries, and that this is associated with reductions in insulin sensitivity, particularly at the skeletal muscle, and a compensatory increase in insulin secretion. And we were interested in really trying to make the direct link between a change in vasodilation and a change in insulin sensitivity at the muscle. So to do this, we also performed a glucose uptake assay at the end of the clamp. So at that point in steady state, we infuse 2-deoxyglucose that gets stuck in the muscle. At the end of the clamp, you then excise the muscle, digest it, and measure radioactivity. And when we did this, we were able to see that 2-deoxyglucose, or just general glucose uptake into the muscle, was reduced after endothelial ARF6 deletion. So the last bit of the puzzle then is, how do you get from dilation to glucose uptake? And the thought there is that it must be changing blood flow to the muscle itself. So in order to assess that, Torkel established a method for measuring skeletal muscle blood flow in our laboratory using fluorescently labeled microspheres. So to do this, he puts a catheter into the carotid artery and the femoral artery, and this will allow for the infusion of microspheres and insulin into the carotid, and then a collection of a reference blood sample from the femoral that's used for the calculation of blood flow. In the fasted state, you infuse one color of microsphere, and then we give insulin, wait 15 minutes, and infuse a second color of microsphere. And this allows us to get a basal and an insulin stimulated muscle blood flow measure. So when we did this, we saw that at baseline, there was already a decrease in muscle blood flow in the endothelial ARF6 knockout animals. In those gray bars, you can see that there was a significant increase in muscle blood flow in response to this insulin challenge that was completely ameliorated in the endothelial ARF6 knockout animals. So putting it all together, we see that endothelial ARF6 deletion reduces insulin stimulated endothelium dependent dilation in the skeletal muscle, that this leads to reductions in blood flow and subsequently to decreases in glucose uptake that lead to a systemic peripheral insulin insensitivity and compensatory increases in insulin secretion. So looking at these two studies together, I think we've been able to show that altering endothelial function is sufficient to modulate systemic metabolism, although I think that the mechanisms that underlie the changes in metabolic function may differ. So in the case of ARF6, it appears that the changes in metabolism are directly related to altering blood flow and nutrient delivery. Whereas in the case of changing mTOR expression in the endothelium, it may be that this modifies the inflammatory and secretory properties of the endothelium, and that is what eventually feeds back to metabolism in the tissues. And so it may be that endothelial targeted therapies could be effective to combat metabolic dysfunction, but that's something that, of course, requires further work. And with that, I'd just like to thank, in particular, Torkel, this was mostly his data along the way, and Shana, who helped with the blood flow studies, Shelby and Sam, who helped with some of the slides in this talk, as well as other members of the Translational Vascular Physiology Lab. I'd like to acknowledge Will Holland, who helped with the euglycemic clamp studies, as well as Matt and Wendy, who provided the 2 floxed animals that we used, and then just acknowledge my funding. And with that, I can take questions.